Hello, everyone. Hi, Clement.
Hello.
Yeah, today, I will be doing presentation about Infopathy, about background of Infopathy, about different researchers who are doing experiments and studies on such phenomena, which we call now digital biology, IC phenomenon, transferring signals from substances to water, storing those signals in the digital way. And in the next presentation, we'll be talking about mobile application, about our database of different signals, different methods on how to use them, different devices, etc. So let's start from the basics. And first, what are ICs? So first of all, a little bit about history. Initially, all the signals recorded from substances we would call informational copies. So IC, it's like an informational copy of medication. Later, we just start using ICs. And also, now we on the platform have not only signals recorded from substances, but also generated signals from different algorithms. But still, IC, it's like a general term for which we kind of use. And ICs are electromagnetic signals that are generated using algorithms or recorded from substances that have healing, therapeutic, or other biological effects. So you will hear a lot of ICs everywhere. That's referring to this type of signals. And whole Infopathy, the platform, the website, is pretty much a database of different those signals, ICs. And talking about a little bit about history of this IC phenomenon. Jacques Benveniste, he was a French scientist, and he was really, really good immunologist. Some people at his time was telling and predicting that he will get a Nobel Prize for his work in the immunology. And one day, one of his colleagues came to him and say, "Oh, would you like to try your system, try its molecules that you study in very diluted way? So dilute your molecule very much time and test it with your system." He was very skeptical because if you dilute molecules few times, so make less concentrated solution, the effect on the biological system should be lower. So, but anyways, he decided to try, and what he found out that yes, initially effect can be reduced, can be go even disappear, but if you keep diluting, it may emerge again. And he even found this like pattern that after some dilutions, it disappears and coming again. You keep diluting, it will disappear and then come again. And he was curious how it's possible that when I place some molecule in the water and then I slowly reduce the concentration of that molecule, so I keep putting more water inside and slowly removing the substance, active substance, effect may still remain. He thought that maybe it's because somehow signal from the substance was imprinted in the water, somehow affected the water as a structure, something. He didn't know what it was. But anyways, he decided to look on all of this from point of view of electromagnetic. That's every substance, it's well known that every substance is unique. It has its own like distribution of electrons and electron levels, etc. So every substance has unique electromagnetic spectrum. And he thought that water somehow can mimic that spectrum. So even if substance is not present in the water still mimic that spectrum. And when you test that water on biological system, it pretty much affects the biological system in the same way as the substance would affect. And yeah, this is pretty much what he did. He diluted substance, and he found out that the effect from just substance with high diluted the water with high diluted substance can have the same effect or similar effect as the substance in normal concentration itself. So then, because he was thinking about this idea from electromagnetic point of view, he did the following experiment. He took electromagnetic coil. Electromagnetic coil in general, it can work as an antenna. If you have some electromagnetic field signal around it, it can pick it up. So the electromagnetic waves are transformed in the coil into the electric current. Then you can amplify that current and then apply it to just another coil. And that coil will emit the signal. It will emit the electromagnetic signal. So when you place pure water inside of this second coil, you expose the water to the signal received from this substance. So he did this thing and he found out that it also works. You don't need to do actual dilutions, like I showed in the previous slide, but you can just transfer the signal to just pure water and test it. And another thing, what you can do, you can just digitize this signal and transfer it no matter what place. Another person who continued some work of Jacques Benveniste and did some experiments was Luc Montagnier. Yeah, he pretty much did very famous experiment when he took signals from the DNA of pathogenic bacteria. He recorded them, stored just in a digital way, and then applied to pure water. And when he placed in the pure water some ingredients to create the DNA, he found out that he can create in this way DNA from which he originally received the signal. So, and that's usually not supposed to be happening. So here you can see it just actually from the very interesting documentary. Can advise everyone to watch. It's called Water Memory, 2014 documentary about Nobel Prize laureate Luc Montagnier. Yeah, by the way, the Luc Montagnier, he received the Nobel Prize for discovering HIV. So he is really one of the best, to work with viruses and DNA, replication DNA, studying DNA, because these experiments require high qualities and professionalism. So yeah, he recorded the signal. He could even analyze spectrum a little bit, and then he informed water with that signal. And he was also using the theory to explain all of this, called quantum electrodynamic theory. We'll talk about this a little bit later. But what he found out again, that when you place ingredients to build DNA to just pure, but water exposed to the signal, you can rebuild the DNA. And usually that's not supposed to be happening. If you just put those ingredients for DNA in pure water, then you just get some random DNA. It will be just randomly assembled. But here, the sequence he received was, I think, 98% identical to the original DNA from which he recorded the signal. So water worked as the matrix to help actually rebuild this DNA. Yeah, it was really, really big experiment. Another researcher who I think also had very good quality experiments, it's Peter Christopher Endler. And he was doing something which also can be called homeopathy or e-homeopathy, electronic homeopathy. It's pretty much similar like to what we are doing. So he was studying the thyroxine and how the thyroxine affects development of tree frog from the larva to the tree frog. And so first experiment, what he did, you just dilute, like in normal way, traditional way, the thyroxine, and then you apply this diluted substance to this small animals, and that should accelerate their growth. And the second experiment, he also did like a dilution, but then he didn't apply directly that highly diluted thyroxine into their and like tree frogs, but he transfers the signal from diluted thyroxine into the pure water and then applied this water to their tree frog in development. And he also found out that that affected their growth. And he did it in two ways. So like in first way, he tried different concentrations of this thyroxine here, because there is dependency. If you have high concentration of thyroxine, it will accelerate their development. If you have low concentration of the thyroxine, it will slow down their development. So first he had high dilution here. He transferred signal from high dilution to pure water. Then he studied how that informed water, imprinted water works, and he found it also accelerates the development. If he, for example, placed diluted very much, like high dilution, and then transferred signal from high dilution into the pure water, he also found out that high dilution slows down the development from larva to tree frog. So it's pretty much like water here has the same qualities as water here. So if you transfer signal from one ampule to another one using the amplifier, etc., you can just get the same thing. You can create that informational copy of the substance. And the question, there's like different theories how it's possible that water can be mimicking that electromagnetic signal. And one of them is developed by Italian scientists. One of them is Emilio Del Giudice, another one is Giuliano Preparata. They were theoretical physicists, and they were studying how a general matter, interacts with electromagnetic fields and condensed matter, most of all. And water also can be considered as a condensed matter when it's like in the liquid state. And what they found out that, like usually we think that water is not organized molecules. It's just randomly moving. There is no interactions. There is no structure. But based on that theory, in just general theory of quantum electrodynamics, if you have two particles, like molecule 1, molecule 2, and they oscillate at some frequency, water molecules, they oscillate. And if you, for example, apply some surrounding electromagnetic field, and there is some condition true, that frequency of this molecule, frequency of this molecule, and frequency of this molecule from surrounding field is similar values, then there will be an attractive force between these two molecules. So they'll be coming together to each other. Now you can imagine if you have like lots of molecules in water and you apply some specific frequency, those many molecules who have the same frequencies, they will be attracted to each other, and they will be forming so-called coherent domains. So this is like a discovery, what was done by Emilio Del Giudice and Giuliano Preparata, that in water there are coherent domains. these groups of molecules that are oscillate at the same frequency, and they just around there floating. One coherent domain can have up to 5 million water molecules, and each coherent domain oscillates at the same frequency. One of the important things that each domain can exist, or like that state can exist for a long time. It's not only like nanoseconds, but even for like hours, days, weeks, months, even they're theoretically predicted. And this is very important, because if water can just remain in the state for like nanosecond, this whole thing doesn't work, because after you imprinted water, it still affects for a long time, and the theory should kind of support that. So if theory doesn't support it, then the theory has something wrong with it. And so if you think about how we imprint this water, our signal usually it's not just like one frequency. Signal from the molecule is a complex spectrum of the signal of the complex spectrum. And like on this slide picture, just also a little bit simplified example. Like let's say we have signal from the substance which has only 3 frequency. Let's just imagine that. So what happening with water when we apply that signal with 3 frequency in it? So for example, we have this like red frequency, just like for simplification, green frequency and blue frequency. So in the water, you have different molecules. Some of them may be oscillating closer to this red frequency. And there will be, again, like if you have signal with different frequencies, you pretty much can be considering it as the 3 different like electromagnetic waves. Yeah, it's like a superposition like phenomena. Yeah, you can kind of break down the signal into 3 parts. So, and they will act like independently on the water or on other system. So let's say we have this red frequency, it will be affecting those water molecules which have similar frequency, and those water molecules will be creating this red coherent domains. Then we have green frequency, and it will be creating the like green coherent domains. Inside of each domain, frequencies oscillating, molecules oscillating at the same frequency, and the blue coherent domains. So here, for example, you can see pink water molecule, like its frequency is not similar with any of those, so it will be not included in any of those coherent domains. And here we can see that because we have those different coherent domains created in the water, so one of them covers this part of the spectrum, and another this part of the spectrum, and this covers this part of the spectrum. So as a result, when we will be considering that water, in general, it can mimic whole spectrum of the initial water molecule. Yeah, so our water molecule had this complex spectrum. We recorded it, then we applied here to the coil, and then we kind of created in the water these coherent domains, different ones, which are in whole together can mimic the whole water spectrum. So as you can imagine that inside of your water, you have like little like emitters, let's say, little like devices almost, which emit different frequencies, like many of them. And when you combine the signal emitted by those like molecules, you will receive the similar spectrum what original substance had. And then the question, okay, we have our water which can mimic the spectrum of the, sorry, I want to actually go back a little bit and mention a few things. Yeah, usually people also ask, well, what is it like effect? What is the difference between effect from the imprinted water compared to the effect from the substance itself? And from what we've seen and from my opinion, even, yes, water forms these coherent domains. Yes, they're stable. They may theoretically stay for like many months, but still they can be destroyed when you freeze the water. You pretty much destroy these coherent domains. If you heat the water to some high temperature, like I think over 40 something Celsius. I mean, the higher the temperature of water, the less stable will be these coherent domains. Because what's temperature? These water molecules will be moving very fast and not going to be able to like stay together, and everything will be just like chaotic and moving. Yeah, the best storage temperature would be like 2 degrees Celsius, because in this way, the water molecules, are, first of all, like close to each other. The water is more dense at that temperature. And also, if you compare effect and just characteristics of this water, these coherent domains, it's still less stable than the substance. So when you play this water, when you drink this water, it would be acting less stable than substance when it interacts with your body. It can be also affected by your body. So it's in general less stable than substance. And there is no answer yet how long water with this state survive inside of the body. Maybe just like 1 minute, maybe like 10 seconds, maybe 5 minutes. Like we don't know yet. But it definitely like less stable than the substance itself. And it seems like also a hypothesis that because of that not very stable properties, this will not have any negative effects on the body as some, pharmacological substances would have, because it's not last long in the body. It's just there for a short time to do the effect. And then, it's not getting kind of collected in the body, stored in the body. It just, after some time, just delivers a signal and kind of goes back to something else, like not these structures, just regular water or whatever the body needs it to be. And then question actually how the body responds to the imprinted water. And here I would like to talk about another scientist. He is not into the water research, but he did very interesting discovery about how our receptors in our body can actually interact with molecules. And he was studying how we smell something. He actually, just go a little bit ahead, he found out that we smell vibrations. According to the traditional view, this is how molecules interact with receptors. You have molecule of the specific shape, and you have receptor specific shape. When your molecule shape matches with receptor shape, interaction happens, and some kind of chain of reactions may start, etc. If it doesn't match, it's not happening. So it's what's called like a key lock principle. Same thing like when you have regular key, you try to open the lock. If you have the same shape, then you will be able to open the lock. If you like wrong key, you cannot open the lock. Like here. And also, So, and but we know that there is like something called electronic key lock already, and maybe this is what happening actually in the cell. This is like a hypothesis that it doesn't matter what actually what shape has this cell receptor, but it's important what electromagnetic signal or electromagnetic spectrum this substance has. Then interaction will be happening based on the spectrum, and cell has some mechanism of reading the spectrum and understanding, how to react on depending on the spectrum. Now, to show how it works, he did 2 experiments. So first, he took the 2 molecules with similar, almost the same shape. And based on the traditional view, we should have same smell. It should have the same smell, these two things. Yeah, but even if they have same shape, they had actually different spectrum, electromagnetic, and they actually had different smell. So one of these molecules smell like cut grass, another is rotten eggs. It's very different smell. So it shows that actually it's not depends on the shape of the molecule. So then he decided to check how will we smell the molecules which have different shape, very different shape, but they had similar, almost the same spectrum. And he found out that they had pretty much the same smell. So it's very cool discovery that our molecules, at least in the nose, our receptors, at least in the nose, can smell the spectrum, like can sense the spectrum of the molecule. It's not important what shape they have, but it's important what spectrum they have. So inside of our nose, we have those little spectrometers, which can interact with molecule based on their spectrum. So here will be like a conclusion, how we interact with their molecule. So here's this electronic key lock principle. Cell receptors response to a substance does not depend on the shape of the molecule, but it depends on its electromagnetic spectrum. And since coherent domains, so if we put them together, kind of mimic spectrum of the substance. So since coherent domains can mimic the substance spectrum, its effect on the cell receptor will be likely that of the substance. So water treated with a signal recorded from the substance has similar effect on the bioorganisms and substance itself. So this is still partially theoretical, but the whole our project is about checking whether that's true and creating the database, allowing people to use like purpose to show how true is this. And next I'll be talking about how this IC phenomenon already been in use, and that's what we're doing is not really new. That's been in use for, like, as I'm aware, for over 30 years, 40 years for energy informational practitioners. Just found some image of kind of old looking device, which kind of showed that it's not from 21st century, but from the 20th century. And and part of this device, so first of all, people practitioners use this device to do diagnostics for person, and then this device has so-called input cup and output cup. So what practitioners would do, they would place in the input cup some substance, like medicine maybe, and on output cup they can place just water, or some people try and test in the sugar beets and other materials. So once you place it here, then they may select some program inside or something. So signal will be transferred from one cup to another, imprinting the substance in this cup. It can be water. Then patient would take it home and use it. And this is just a sample of some a more modern device, also for diagnostics and something else. They also have maybe even databases inside of different signals. So practitioner would put just some tincture inside, select the program and transfer it. And what we're trying to do, just kind of bring everything into using internet, bring everything in the online space, whole database into the online space where people will be able to, or already able actually to select different signals from different substances and prepare their own water at home. The whole development, here I would like to kind of say in the same time, like, thank you for everyone who used Infopathy and for their suggestions, feedback, because pretty much development of Infopathy is like crowdsourced. We initially just started building it when people were requesting, do like, for example, create like this feature or make this available. So it's pretty much like a collective contribution on making it, and everything also like stimulated by the user's feedback. So it's kind of exists because like people use it, people like it, and kind of people pushing it forward. It's available right now via the browser. You can just go on your phone, on your computer, to the infopathy.com and go to the main catalog, search through this. We'll go into more details how to use it in the next presentation. And thank you very much for your attention. If you have, Clement, if you have any questions, maybe regarding the material talked about, you can ask me. If not, we can move to the next presentation.
Very good, Anton. Thank you very much for your time. It's mind-blowing. It's a very good explanation of what's going on behind the scene.
Excellent. Thank you very much.